This video is going to look at the exceptions to Mendelian genetics. Before we start looking at these exceptions, we'll review the rules of Mendelian genetics. So some of the key elements of Mendel's original model were that heritable traits are determined by heritable factors called genes. And we know that genes come in pairs. The second thing, genes come in different versions called alleles. There's a dominant and the recessive allele, and we know that the dominant allele always masks the recessive if it's present. Number three, during gamete production, each egg or sperm cell receives just one of the two gene copies present in the organism. And number four, genes of different traits are inherited independently of one another, which is the law of independent assortment. Okay, and you would have learned about that with meiosis as a source of variation. Okay, so this is usually the case with Mendel's genetics, but there are some exceptions that actually gives rise to some different combinations of alleles that gives off different characteristics or the phenotype. Okay, so these are the four we're going to talk about today. So incomplete dominance, codominance, multiple alleles, and lethal alleles. Okay, so the first one is incomplete dominance. This is known as the blending of traits in the heterozygote. So usually the case would be if we have, for example, a red flower and we cross that with a white flower, the dominant alleles are going to have more of a say. They're going to be expressed in the phenotype. So all the flowers should be red if we cross red and white together. This is not the case with incomplete dominance. We have a blending of the traits. So red and white would make pink with the flowers. Okay, it's pretty similar with the Labradors down here, brown Labrador, white Labrador, and making that golden color in between. Okay, so it is where heterozygous individuals are a common of both of the dominant and recessive conditions. Okay, I just want to point out here that with the flowers, one white, one red, and two pink. So the phenotypic ratio is going to be 1 to 2 to 1, red to pink to white. And here is one other example, looking at flowers again, just looking at these different letters, whether they are red or they are white in the color. 1 to 2 to 1 ratio. All right, the second type is codominance. Now, codominance occurs when a single gene has more than one dominant allele. A heterozygous individual will express the phenotypes associated with both alleles. So it means that one's not going to be expressed over the other, or there's not going to be a blending, but both are going to be expressed at the same time. So you may have your dominant and your recessive alleles both expressed. Okay, we can see here we've got a picture of a flower again, but in this case we've got pink and we've got white. So both the pink alleles and the white alleles are present and expressed in the phenotype. Okay, roan cattle is a good example. And also we've got some chickens down the bottom here. Okay, speckled is a combination of those two. Okay, so the third out of the fourth is multiple alleles. Now, a gene can have more than two different alleles. The genes responsible for human blood is an example that has multiple alleles. To further complicate matters, some of these alleles show complete dominance and some of them show codominance. Okay, so there can be a bit of variety here. Okay, so basically to explain that in a little bit more detail, multiple alleles, we talked about blood type. Okay, it's basically to do with the carbohydrates that are present on the outside of the red blood cell, or the surface proteins. Okay, all of these red circles, I guess, are the red blood cells. Okay, and they have the A's, the B's, the A mix between A and B, or no surface protein. Okay, and that actually determines what blood group you are. So single gene has 4 phenotypes. So type A can be AA or AO, type B, BO, AB can be only AB, and type O has only OO, so basically no carbohydrates. Okay, so here's another example, just with the blood type here. Okay, there is a mixture here. No carbohydrate, can have 2 of those, 1 of that type and 1 of that type. And these are the different alleles that are present, or the genotype. And we're really interested in the genotype because this is genetics. Okay, so genotype as the A carbohydrate can either be A or a mixture of A and O, as we can see. Type B is B, or I can have a 1B and 1O. And then AB, okay, is both of those there, and then O is nothing, which is the recessive. Okay, so the last one is lethal alleles. Now, lethal alleles cause the death of an organism. They can be dominant, incompletely dominant, or recessive. They usually arise due to a mutation or a change in an essential gene. The gene cannot produce a functioning version of the protein, and that causes the organism to die. Okay, so here we've got some pictures down below of lethal alleles and how they can affect these organisms. So basically, causes of death of the individual can be dominant, incompletely dominant, dominant, or recessive. Okay, so pretty much what we've just said before. A good example I like to talk about is with these mice for lethal alleles. Now, we've got 2 different types of mice. One of them is the brown mice, which is the agouti, and then one of them is the yellow mice, which is the yellow color. Okay, now if we crossed these together, so a heterozygous individual, so big A, small a, with big A, small a, if we have a white colored mouse that is born, that is the lethal allele in this case. Okay, and that means that this mouse is dead. Okay, because the lethal allele, you cannot have a white mouse, or it is detrimental to the mouse. So if this was the case, okay, we're going to have 2 of the agouti brown mice and 1 of the yellow. So what this does is this actually changes our ratio, our phenotypic ratio. If we look up here, and says there is a 3-to-1 phenotype ratio. Okay, but one of them dies. So if it dies, we're going to be left with the 2-to-1 ratio. So 2 brown to 1 yellow. Okay, so that basically covers all of the 4 exceptions to Mendelian genetics. Again, these were some of the learning intentions if you wanted to look through some of these ones. All right, thanks for watching.